Spalding, and I work for the EGA at the European Bioinformatics Institute here in the UK. And I'm going to talk about ELIXIR and IMI OncoTrack and how we've done a scoping study to provide long-term data storage and data sharing for this OncoTrack data. So I'm going to talk about the data challenges. So we know that the number with the development of sequencing technologies and associated reduction in the price of sequencing, the number of data production sites have been increasing across Europe. This in itself then increases the amount of data that's available within the life sciences. Here you can see how data growth has gone up over EMBL-EBI. And this creates issues, especially with respect to human data. So human data, obviously, we have privacy concerns, we have data protection legislation to deal with, so we need to deal with secure access and how we govern the access to these data. But at the same time, we have to deal with the open data mandates of national and European funders, such as the Wellcome Trust, the EU, and so on. So this talk where we're looking at the study of trying to improve data access for consortia data using IMI OncoTrack as an example. Here's an outline of the talk. So I'm going to talk about EGA to start with and how we could use EGA to facilitate this. And I'll go through the statistics, the metadata model for EGA. I'll then talk a bit about the IMI OncoTrack project, give you a quick overview, and then detail some of the data collection, the data types, and the infrastructure that they've been using. And then I'll go on to the study itself, the aims of the study, and the results of the scoping study on how we can get this data and improve the access to it in future, and give some conclusions at the end. So the European Genome Phenome Archive, or EGA, is a joint service which is run by the EBI and the CRG, Centre for Genomic Regulation in Spain. And it's a secure permanent archive for all types of genetic, omics and phenotypic data in humans. Now, crucially, all the data in the EGA is consented for research use. It's under some form of consent agreement, which I'll talk about in a bit. And this means that all the data access is managed, so you have to apply for data access, and all the data is encrypted when it's delivered to the requester. And the submissions, we have a very large range of, diverse range of submissions, which include genome sequence, transcriptome, and epigenome experiments. We have cool variants and genotypes, and we have a lot of sample phenotypes as well. So what is controlled access data? Well, this is where an informed consent has been signed by a patient or individual specifying the controlled release requirements for their own particular data. So therefore, all this data is obviously human, and for EGA, it has to be personally identifiable data types. And this can be raw data such as FASTQ, BAM files and so on, processed data, for example, the VCF files and called variants, and phenotypic data, so it can be certain rare disease information or age and so on. A lot of the data at EGA is affiliated to biomedical research or consortium projects. I'll give you a list of those in a bit. And the access, again, is by formal application. And the people you apply to are the data access committees, and these control the access to the data. So the EGA, why does the EGA have such a diverse range of data? With open access data, you have specific archives that tend to deal with specific types of data. For example, sequence data goes to ENA, variants go to EVA, structural variation goes to the DGVA. The thing that keeps all the data in common within the EGA is the fact that it's all under controlled access. So just keep in mind, this is why we have such a wide variety of data within the EGA. So some context and metrics to the EGA. The EGA has been going on for about 9 years, and its role is a secure archive for the controlled distribution of consented data. We actually have almost 9,000 data access accounts now, and over 450 submission accounts. So these can either be consortia or individual lab laboratories. Currently, we have just over 3.7 petabytes available of data available for download, and this is in just under 3,000 data sets. And last year, we distributed 3.8 petabytes of data to various requesters. And we have a help desk, and we have around 200 contacts with the help desk a month, and the help desk is also quite important in regards to this project, so I'll explain in a bit. So, as with the first slide, you can see within EGA, it mirrors the kind of data growth you see with all sorts of life science data, especially genomic data. Also in EGA, we have loads of different technologies, so this is demonstrating some of the diversity of the data at EGA. We have GWAS studies, exome sequencing studies, genotyping, sequencing, and so on. We also have quite a range of study types, but most of them are actually to do with cancer, although we have cardiovascular, inflammatory, and so on disease studies. And in terms of cancer, the most popular study type is actually on blood cancer, followed by breast and skin. In terms of download from the EGA, we've been running a secure download service, and that distributed 3.8 petabytes last year. And again, you can see the growth here, which is basically doubling in size each year in terms of requests and volume of data accessed. In terms of locations, the EGA delivers data all over the world. You can see here, a third of our data has been going to the USA, but you've got the UK, all of Europe, some, well, actually 0% in Australasia, apparently. And in terms of submissions, we have over 200 institutions which will submit to the EGA. These are basically represent every continent apart from Antarctica and South America. And we have big consortia who submit. So the yellow points here on this map are actually ICGC sites that are submitting to the EGA. And here you can see some of the other consortia that submit to the EGA, not only ICGC, but we've got the Wellcome Trust Case Control Consortium, RD Connect, Institute of Cancer Research, and so on. So I'm going to describe a bit about the EGA metadata objects now, or later on in this presentation. So the EGA tries to synchronize its metadata object requirements with the ENA and SRA, which both take in non-controlled access data. The reason being is that it means the data models for submission to either controlled access or open access archives should be similar, if not identical. And this facilitates the bioinformatic pipelines to submit data to these different archives. So we have a study and project object which contains the information about the project and the study. We also have the samples, and these have attributes which describe the sample, for example, the donor, the gender, and the phenotype. We have experiments which describe the experimental methodology used and the platforms and so on, and run and analysis objects. Now these two objects reference either raw data in case of a run, or process data in case of an analysis. And it's to these objects that the files are attached. In terms of EGA, we have a set of extra specific requirements to deal with the controlled access. So we have the Data Access Committee, which defines the Data Access Authority, and the Policy of Data Access Agreement. This is basically the terms and conditions which are applied to someone who's requesting access for the data use, and the data set. And this is a package of files into units which are used for data access and distribution. So a single policy will apply to a single data set. Here's a diagrammatic diagram of the metadata at the EGA. You can see in blue, these are the objects that hold the files, the runs and the analysis holding read data, underlined or analyzed data. You see the data set that links these two together, and the policy and the DAP, and then the experiment samples, study, and so on, which describe these data. EGA Accessions, so it gives stable identifiers to most of these objects. The most important ones are the study of project, which is an EDS ID, the sample, which is an EGAN, and probably the Data Access Committee actually after that, which is an EGAC. So the metadata at EGA has been stated since 2013 as being public. So this is to facilitate data discovery. If you want to find out what data is in EGA and you don't have access to the data, it needs to be public so you can query to a certain level the data within EGA. So the object attributes, and in terms of samples, the donor ID, gender, and high-level phenotype are all stated as being public and are queryable via our website or REST APIs. For low-level phenotype data or consented sample metadata. These are currently archived as a file, which ensures they're under controlled access, so they're not publicly accessible. Examples would be, for example, age here, or actually geographic location and so on. So a typical EGA submission consists of one study which should be linked to multiple samples, and with each sample linked to a separate experiment and a run. And these tend to be linked to a single data set with a single DAC and a single policy being applied to them. So how do you access data at EGA? You have to apply to a DAC. And so the DAC, if you look at the diagram here, the DAC actually applies data access agreements for data sets. So here in the diagram, there are two data sets, the red and the green, which have different data access agreements or data use conditions applied to them. And we have three researchers on the other side, and you can see one researcher has got only access to red, one researcher's only got access to green, or one researcher's got access to both. And if we see this on this diagram here, you can see the whole process of access to data sets where a researcher requests access directly to the DAC and not to the EGA. The DAC themselves then creates either an account or grants access to an existing account for that user, and then that user can download those files and analyze them, perform analyses on them as they see fit. So why do people submit to EGA? Why do we think it's important that people submit to EGA? Well, it maximizes the benefit from the data. So we distribute it for you, and we deal with the controlled access and so on. Funders and journals now requiring researchers to have a data sharing policy. The examples of this would be the Wellcome Trust and their policy on data management and sharing, but also journals and so on with Nature and PLOS also having policies in terms of the availability of data and materials. The other advantage of submitting to EGA is you can utilize our distribution technology and access tools. You've seen earlier on in the statistics the amount of data that not only we archive but also distribute on behalf of other people. And ultimately, this makes the data at EGA fair. So we help make data findable, so by giving it permanent identifiers accessible, we provide the technology to download it securely. Interoperable, we have the APIs which interact with other archives and so on to allow the data to be pulled down and reusable, again with the identifiers and the fact we're using a long-term archive. So moving on then to IMI OncoTrack. Well, the IMI is a large public-private partnership for life science between the European Federation of Pharmaceutical Industries and Associations and the European Commission. And OncoTrack itself is an academic industry translational research project which had the goal of identifying biological markers that help with the understanding of tumours and the relationship between biological heterogeneity and the tumour variation in response to treatment. Now, the OncoTrack project has been funded for a specific timescale, so the question here is what happens to these data after the project has ceased, and how we maintain access to it and distribute this data so we maximise the benefit of this. And the data within this includes phenotype data, genome sequencing data, computer modelling data, imaging and xenograph data, and also the transmark subset of an OncoTrack database, which we'll talk about in a minute. So OncoTrack, what's its purpose? What is it trying to do? Well, apparently drug development frequently fails, as does biomarker validation. And despite extensive research, there's been modest impact on the precision medicine or the benefit to specific patients who've been sequenced from all this. For example, approximately 200 pharmacogenomic biomarkers are listed in the FDA labels for licensing drugs, and 50% of these are for oncologic indicators, but only a small number are currently being selected for use as a companion diagnostic. So the premise was that the source data does not represent the full complexity of the tumour well enough to allow identification of novel biomarkers for the stratification of cancer patients. So in effect, there's a lack of data or lack of necessary data to describe and allow determination of and identify the correct biomarkers. So the strategy was to create a new methodology of data collection, which allows a more complete or complex data set to be used for biomarker discovery and validation. And in doing so, this would mean surgical specimens of tumour, metastases, blood and normal tissue are all collected at the time of a patient diagnosis, and the patient will be followed up on samples taken in case of the progression of disease. So the data that was collected, there was tumour sample sequencing, and this was genome, transcriptome and methylome data. The tumour sequences were compared to normal liver or colon tissue or germline DNA from peripheral blood lymphocytes. Xenograft and organoids were derived from these tumour samples, and these were used to assess drug response as transcriptome analysis and confirmatory mutation analysis was performed. Proteomic mass spectrometry measurements, reverse phase arrays and proximity ligation assays were used, and conventional bioinformatic approaches were also used, along with systems biology and an in silico modeler. So you can see there's a huge heterogeneity of data here, a very diverse range of data that's been collected. In terms of data management, and this also relates to the EGA in terms of the data is pseudonymized, so it's under data protection legislation, so it does need controlled access. And also, by pseudonymizing the data, you can then feed back results to patients. There's quite a large volume of data that's collected, and as I said, it's very diverse, and both in terms of the data sets and the formats. And analysis is performed over these diverse resources. So here's a diagram of the data infrastructure. And the clinical data is stored in an open clinic database. And this is the clinical data, patient data, and treatment data. And this is hosted ELIXIR. Some of the selected data is then uploaded into the OncoTrack database. And these include the analyses performed on the samples and blood samples and tumour cells. And as I said earlier, these are next generation sequencing, proteomic analysis, and so on. A subset of the data from the OncoTrack DB and OpenClinica DB was stored within the Transmart with data warehouse, which was hosted by the other IMI project, EGA. So the study that we're talking about here, this scoping study, was to try and begin to understand how we can perform long-term management requirement on both the data storage and data governance and access and distribution of data produced by the IMI OncoTrack project in future after the funding for the project has ceased. We also had to look at how we're going to map the metadata. You've seen how complex and diverse the data the IMI OncoTrack project collected, and how we can map that to the metadata within EGA, and where the difference is between the public and private data, so the data that's purely consented for private or controlled access, and the data that's public use to be used to discover these data. And in doing so, this is hoping that we would now generate a use case for other IMI consortia, and actually, in general, other consortia to submit their data to the EGA and allow the EGA and hence ELIXIR to provide these data throughout Europe. So so we know that the data from IMI OncoTrack is anonymized data, so it's under consented access, and the consent actually is only for oncology research. And so to perform long-term data distribution, we have to provide a DAC who will to whom a requester will apply, and the data access agreement or data use conditions will be applied to see whether their use of the data conforms to that, and if so, they'll be granted access. But the scenario here is that if you don't have a DAC, then there's nobody to apply the data access agreements. If there's no data access agreements, there's actually no access to these data. One of the options considered during this was to actually use the EGA itself to apply data access agreements developed by OncoTrack to people requesting this, so the OncoTrack did not have to provide their own DAC. But in the end, it was decided that OncoTrack would create their own DAC, which is now in place. They've also developed their own data access agreements, and these are currently under review to ensure they conform to legislation throughout the EU. We estimated the costs of this, so both for OncoTrack and any other use cases, we need to know the cost of the DAC, and how expensive administering these DACs and data access would be. And to do that, we went to the Wellcome Trust Sanger Institute, who run a large DAC for a lot of their studies through EGA, and we asked them to estimate how expensive effectively it was to administer a DAC on data sets. As they have quite a wide variety of data sets, they came back and they said that each access was roughly equivalent in terms of man hours to apply. So the main effect was actually how often access was requested to a data set. So a higher frequency data set would require more man hours than a low frequency data set. Their data has to be taken with a certain has a certain requirement in terms of the fact that these stack is controls access to a load of data sets, so it's highly accessed, so they have some level of automated systems. They also have specific people hired to run and allow access to these data sets. So the values here of, I think, 2 hours per request are specific to them, but it does illustrate how we would expect the cost to be distributed for any other DAC at all. In terms of hardware costs, at EGA we calculated these were minimal due to the relative size of the OncoTrack data to the EGA. The OncoTrack data is less than 10%, less than 1% of the size of the EGA currently, and OncoTrack are expecting to submit to us in 2018. Given the growth of data, it would be less than 0.5% of the size of the EGA by the time they submit. So, and as time goes on, the proportion of data that's OncoTrack within EGA will be minimal. So those costs are very small indeed. So the other thing we looked at was the data upload to the metadata mapping. And here you saw, specifically with OncoTrack, you saw how complex the data was. And so we need to make a unique mapping per submission to the standard metadata model that the EGA uses. And to do this, we need to use the EGA help desk and the submitter both working together to map these metadata. Here is an example of how we were trying to do this. So for example, this is a spreadsheet, and we have on the left-hand side some of the tables or objects within OncoTrack that were to be submitted to the EGA. You can see on the color code that some of them were not going to be imported, some of them were going to be archived as a file. This means they're under controlled access, and some of them will be stored under the file metadata, so that they were at higher level and associated directly with an object. So you can see the object column in column C, these were the objects the metadata would be associated to. And this actually is another cost on pulling data into the EGA, because this has to be done by humans. There's no way of automating this, because when you start to submit the data to the EGA, existing projects will not have mapped their data to the EGA metadata model. So this will need intervention both by the EGA help desk and the submitter themselves to map these data and determine which parts of the data go public and which parts of the data go private. Currently, we estimated that so far the EGA help desk has used around half a person's month and time on doing this, and the submission actually hasn't started, so there'll be more work to do once the submission starts. Also, we're looking at how areas within the EGA where we don't have adequate coverage, basically, for submissions such as this. And one of these examples would be the sample and also the SOP for importing these data. So we're developing an SOP. This is using an existing EGA help desk person to help work with submitters on a one-to-one basis to map their data, and how we map that data into the EGA, and how they set up the DAC processes, any legal requirements they need to go through, and so on, and try and improve the sample data representation. This diagram here at the bottom of this slide, this is an example of the samples collected from a patient within the OncoTrack project. There are 3 separate samples and 5 technical replicates here, which is why it's, there's so much to get on the slide. Here's a simpler version of it. And the EGA currently does not represent this well at all. We only have a single sample object here. We would like to have an individual patient object check from which samples are derived, and we also need to be able to deal with technical replicates of samples and also how we run these in time series analysis. So earlier on, we were talking about how we're measuring drug responses, so we need to actually measure how a sample reacts to certain drugs over a period of time. Also, in addition to that, we also need to store under controlled access both cell cultures and xenograft data, again, which occur within the IMI OncoTrack project. Also, the IMI OncoTrack project uses CDASH as a way of collecting the clinical and patient data, and so the EGA needs to improve its ontological or standard support for this. Currently, attributes within a sample on the EGA are not specified by ontology or any particular standard. So to improve the data discovery, whether it's at the public level or indeed programmatic access, once you've gained access to the data after controlled access, we need to be able to manage the ontologies better. We need to be able to map these ontologies better so that we can then start performing cross-queries across both, for example, an OncoTrack submission and other studies within the EGA. And we also need to make sure we make a clearer specification of the difference between the controlled access attributes and public access attributes. For example, and these aren't OncoTrack attributes, but for example, controlled access attributes such as date of birth or geographic location, which we've seen are personal data, identifiable, and so therefore under controlled access, must be kept under controlled access, whereas other attributes such as, for example, sample site, for example, liver or colon, or high-level phenotype, for example, breast cancer or blood cancer, for example, those could be public and should be public to help enable data discovery. So in conclusion, this study has helped in us developing the partnerships with industry to access these data and to make sure this data is made accessible in the future, and we provide the access to this data after funded projects have ceased. We're developing and created SOPs to facilitate data load at the EGA, and we've made cost estimates for that. It looks like the EGA is useful to provide long-term access to translational data for the community, and there are potential benefits to small and medium enterprises here. So by the EGA and ELIXIR providing the infrastructure, data infrastructure here, and the archival processes, plus the distribution processes, there is a significant cost savings for small and medium enterprises who perhaps don't have the resources to do this themselves. In terms of the costs, the prime costs are for the metadata mapping on data upload, and then the DAT costs the ongoing DAT costs to ensure data access. But the hardware and storage costs are minimal, as you've seen, due to the volume of data within EGA, with the obvious caveat that the volume of data submitted is not a significant percentage of the EGA data, or that the data submitted doesn't require any special distribution or access processes, which EGA doesn't currently support. We've also need to improve the metadata in the EGA for translational data. You've seen how complex the sample data from IMI OncoTrack is. We need to be able to better model that so there's a clearer distinction both between public and private attributes, and it's easier to interrogate these data. And this will also benefit other consortia and existing consortia, such as RD Connect and so on. But the most important thing here is that we're actually making all this consortia data fair, findable, accessible, interoperable, and reusable, and it should remain so going forward. So we maximise the usage of this data and the scientific benefit that the collection of this data has. So finally, I'd just like to acknowledge these people from our collaborators at CRG, with the EGA, Geordie and Dowd, Alexander and Jeff here at the EBI, David Henderson at Bayer Healthcare, Riha at Alakris, and Susanna and Nicholas Serena at the ELIXIR Hub. Thank you, and do you have any questions?